Welcome to part 2 of online lecture 8. In this section I'm going to detail the steps involved in the Southern blot and PCR methods that are going to be used in the CASET study on Huntington's disease that we'll be using in class on Wednesday. In part 1 of this lecture I mentioned that the Huntington's chorea was caused by a mutation in the Huntington's gene, and that those mutant proteins led to death of brain cells and neurological symptoms that resulted from them. And I also mentioned that the precise genetic mechanism that was responsible for the disease is known, even though all of the steps that actually result in the phenotype are not very well understood. And you may recall that that genotype had to do with an expansion in the number of CAG repeats that occurred in the Huntington's gene. And this slide is actually showing the effect of the number of CAG repeat units on the onset of, on the age at onset of the neurological symptoms in Huntington's disease patients. And one of the things that you'll notice is that the disease actually becomes much more severe as the number of CAG repeat units increases. And what I mean by severity in this case is actually at the age of onset. These data have a very steep negative slope here, and you can see that when individuals have, really more than about 60 CAG repeats on one of their chromosomes. Remember, this is a dominant condition. And when they have more than 60 repeats on even one chromosome, that the age of onset is actually under 20 years old. You'll also notice that for individuals with less than 35 or so repeats, that they're not included in the Huntington's disease patient pool because those individuals actually are normal and don't have the disease phenotype. So what this shows us is that if there is a way to determine the number of CAG repeats that an individual has, you can actually predict whether they're going to develop Huntington's disease or not. And this is the theoretical basis for the CASET tutorial that we are going to do in class. So there are two different procedures that can be used to determine whether or not an individual has Huntington's by looking at the number of repeats of these CAG repeats that they have on their chromosomes. So we're going to look at two methods for focusing in on specific DNA fragments out of all of the fragments in the genome. And in this case, both methods are going to focus on this Huntington's gene. And both methods actually allow us to determine the size of the fragments in question. And of course, the size of the fragment would be important because individuals who have chromosomes with a large number of these repeats would actually have much larger DNA fragments because they've got this 3 base pair region that's repeated over and over again. For example, if you look at one of the individuals on the bottom part of the screen here, around 100 CAG repeat units, that would be a piece of DNA that would be 300 base pairs longer than, say, someone who had, say, no CAG repeats, or for someone who had around 20 CAG repeats, the difference would be more around 240 base pairs. So if you can actually determine the size of an individual's Huntington's gene, we can actually, and the number of repeats that they have, we can actually predict whether or not the individual will have the disease phenotype. And so in the case that we're going to do, we are going to run a Southern blot and then a PCR analysis of individuals in a family that have been affected by the disease, and we'll see what the results say about the likelihood of the as yet asymptomatic people for developing Huntington's disease. We're going to start with the Southern blot, and this slide shows an overview of the Southern blot procedure. which basically involves taking some DNA fragments and placing them on an agarose gel and separating them by electrophoresis. That procedure was also explained in the lecture on great experiments, and so I encourage you to go back to that if you need further explanation of how gel electrophoresis separates DNA fragments according to size in detail. What I'll tell you, just to help jog your memory a little bit, is that the samples get loaded in the top in what are called wells, and these are just little indentations that are left in this jello-like substance by a mold. It's called a comb that you stick down into the gel, and it makes these little wells. So when the gel hardens, you pull the comb out and it forms these little pockets. And in a southern blot, you load your DNA samples into each of those wells, and then using electricity, you basically force the fragments to move through the gel, with the smallest ones moving the fastest because they encounter the least friction. And in all gels, it's vitally important to add a size ladder, and this is just a series of fragments that you already know the size of that you can use to compare your samples to. So this is the first step in a southern blot, is to generate these DNA fragments and to run them on a gel. The DNA fragments are generated by using a procedure that's called a restriction enzyme digest. And a restriction enzyme digest is a procedure that actually uses an enzyme that has been isolated from a different organism or produced using recombinant DNA techniques in a biotechnology laboratory. And restriction enzymes are really fascinating, and a great example of something that happens really often in molecular biology lab techniques, and that's when we discover what something does in nature and we co-opt it for laboratory uses. So in nature, restriction enzymes actually serve as a defense that bacteria have against invading pathogens, particularly bacteriophages. You may also recall from the Great Experiments lecture the Hershey-Chase experiment, where they used radio-labeled DNA and protein to try to determine whether it was proteins or DNA that were the hereditary information. The key part of that for understanding how restriction enzymes work is that you may remember that the way that the bacteriophage attacked the bacteria was by injecting its genome, which can be DNA or RNA, into the bacteria. And literally what happens during a viral infection is that the bacterial RNA polymerase and ribosomes actually start transcribing and translating viral DNA and viral proteins. So it literally is a molecular hijacking of the cell. And so what are those poor defenseless bacteria going to do? Well, what's happened is that over millennia there has been natural selection for bacteria that have some way of combating this kind of invasion. And that mechanism is what's called a restriction enzyme. So in nature, these restriction enzymes actually target foreign sequences, and they're kind of like, you could think of them like molecular scissors. So the job of a restriction enzyme is actually to recognize DNA, foreign DNA specifically, and chop it up into a bunch of little pieces. And you can see how it would be important for bacteria to find a way to protect themselves from their own restriction enzymes, which they do by the addition of some methyl groups to their genomes. But that discussion is outside the purview of what we're talking about right now. So restriction enzymes then are literally made for cutting DNA up into a bunch of little pieces, and that's what they're used for in the southern blot procedure. But restriction enzymes are specific. They don't just cut DNA into however many pieces and however many places. Each restriction enzyme recognizes a very specific sequence of DNA. And the example that you're looking at on this slide is of a restriction enzyme that's called EcoR1. The eco part comes from Escherichia coli or E. coli. That tells you that's the bacterium from which this enzyme was initially isolated. It was from strain R, so that's where the eco R part comes from, and then 1 just indicates that it was actually the first enzyme that was isolated from that strain of E. coli. So eco R1, like all restriction enzymes, has a very specific target. It only cuts the DNA sequence that you see here. So it cuts when there is a GAA-TTC restriction, sequence on the target chromosome. We call this a restriction site or a recognition sequence, and it only cuts this particular sequence, and only very specifically between the 5' guanine and the adenine that follows it. You'll notice that when you look at the EcoR1 restriction enzyme, that if you look at the complementary strand, that it also has the same sequence. So if I start here, you can see guanine, adenine, adenine, thymine, thymine, cytosine. And so these restriction enzyme sequences are what we call palindromic. So they actually, the sequence is the same on both the forward and the reverse strand of the DNA. And this is important so that we have double-stranded cleavage of the DNA. And this is true of most restriction enzyme recognition sequences. So the first step of the southern blot is going to employ this restriction enzyme digest technique, and this is basically being done to cut an entire genome, which is enormous. A human genome is enormous, and this is going to cut it up into a bunch of little pieces so we can actually work with it. So we can run it on a gel in an hour or a little more than an hour and actually see the size fragments separate. If you tried to do that with an entire chromosome, it would move very slowly, and it would just be very difficult if you could even find a gel with a pore size large enough for that to move through. So the first step is going to be a restriction enzyme digest of the DNA fragments. And prior to that, there has to be some form of isolation of the DNA from the cells as well. But I'm going to talk about that procedure. So once the restriction enzyme fragments have been created by a restriction enzyme digest, and by the way, this is typically done by taking a restriction enzyme and a buffer and mixing those together and adding some of your DNA sample that you're working with. And you mix these together in a tube, and typically they're incubated overnight at about 37 degrees Celsius, which is the optimal temperature for restriction enzymes, particularly that are isolated from E. coli. And 37 degrees Celsius is about the average body temperature of a mammal. And so E. coli are what are called coliform bacteria that inhabit the colons of humans. So it's not surprising that their optimal temperature would be near body temperature. Anyway, the restriction enzyme's done overnight, and then in the southern blot, the fragments would then be loaded onto the gel, and you would have one lane or one well per sample that you were interested in, and then this sample would be allowed to run. The next thing that would happen would be that the fragments would actually be blotted onto what's called a nylon membrane, and that's what's being shown here on the top. And so this is literally, it's almost kind of like making taking an ink stamp and taking the ink and placing it on a paper, although in this case your stamp is your agarose gel and the paper is this nylon membrane. And so now you're not working with the gel anymore, you're working with this nylon membrane. And still, at this point, even though this slide shows you what looks like it would be individual bands, if you had actually digested an entire genome, a human genome, with a restriction enzyme and ran it on a gel, you would just have smears in all of these lanes because you'd have so many different fragments of so many different sizes. You wouldn't see any discrete bands on it at all. So the purpose of the southern blot is actually to try to select out only a few very specific samples from a large pool of samples that have been run on the gel. And the way that we do this is by adding what's called a labeled probe. And the probe in this case would be the probe for the Huntington's repeat. And the probes are shown here in yellow. And they're shown yellow because they have some kind of label attached to them. And we've already talked about labeling in the Hershey-Chase experiment. We've talked about using radioactivity during the measles and Stahl experiment. And we also talked about labeling in the example on the beta cells in Doug Melton's work on trying to find a cure for diabetes. So this label could be radioactivity, it could also be a fluorescent dye. And typically this probe is allowed to hybridize to these samples, usually overnight. And then the membrane is washed to remove unbound probe. And what should happen is that you should only have the probe appearing where there was a DNA sample that was complementary to it. And so I forgot to mention that there is a denaturation step where the double-stranded DNA is separated before the gel is run, so that you're looking at only single-stranded fragments. So your probe then is another single strand of DNA with a label attached to it, but it's only going to attach to fragments that contain this Huntington gene and the repeats. And once the probe has been applied and the excess unbound probe has been washed away, the sample is then exposed either to X-ray film or some other means of detection to look for DNA fragments of certain sizes in the various lanes. So we're going to look at a short animation that actually gives you a dynamic view of this process, which I hope will help improve your understanding.
The southern blot is used to verify the presence or absence of a specific nucleotide sequence in the DNA from different sources, and to identify the size of the restriction fragment that contains the sequence. In this procedure, the DNA is isolated from each source and then digested with a specific restriction enzyme. The DNA restriction fragments are then loaded onto an agarose gel and the fragments separated by electrophoresis according to size, with the smaller fragments migrating faster than larger fragments. The fragments are made visible with ethidium bromide. The DNA is then transferred from the fragile gel to a nylon filter. Next, the radioactively labeled nucleic acid probe is added. The probe binds to complementary DNA segments. Note that the DNA segment being probed is not present in organism B. To detect the position of the radioactive probe, the nylon membrane is covered with an X-ray film. After development, the positions of the probe become visible.
Okay, so in the case of Huntington's disease, we wouldn't be looking at 3 different organisms. You'll be looking at 3 different members of the family. And all of them should actually have some number of copies of the Huntington's gene. But what we'll be able to do is actually figure out how many copies they have, and therefore how likely they are to develop the disease. So the southern blot is one way of probing for a specific DNA fragment and then determining its size. Next, we're going to look at another technique that can be used to do the same thing, and this technique is called the polymerase chain reaction, or PCR. The polymerase chain reaction is a really simple and elegant technique that was developed by Kary Mullis when he was working for a company called Perkin-Elmer. And Kary Mullis is an interesting dude. He actually won a Nobel Prize for this development, and has also been a very outspoken advocate for the use of LSD. So interesting guy. Anyway, Kary Mullis did something very important because he developed this technique which allows us to make copies of DNA from a very small starting amount and literally making as many copies as we want. And he did what I've already described with the restriction enzymes, in that he basically just co-opted a process that already occurred in nature and put it to use in the lab. And the key to what Kary Mullis did was to find an organism that had very heat-stable enzymes that could withstand being heated and cooled repeatedly without denaturing. And of course, the way that he did this was to go someplace that's hot, and he went to a bacterium. So Thermus aquaticus was a bacteria that was isolated from hot springs, and so obviously you can see how this species would need to have heat-stable enzyme. So Carey Mullis used this enzyme and developed a laboratory procedure which is basically just a series of reactions that heat and cool DNA in the presence of this enzyme, this DNA polymerase that was isolated from Thermus aquaticus. It's called TAC DNA polymerase. The T comes from Thermus and the AQ comes from aquaticus. And Carey Mullis basically replicated in the lab the DNA synthesis technique. He provided a little primer. Remember, DNA polymerases require a primer to attach to. And for our purposes, this primer choice is going to be incredibly important because the primer choice is actually what dictates which part of the DNA is actually amplified or copied using PCR. So those primers basically are like they choose the target DNA. They're sort of like an arrow going for a bullseye, and only the region between the primers will actually be amplified. So in the case of Huntington's chorea, it would be important to use primers that were known to target the Huntington's gene, and that's exactly what's done. And then once the PCR process is over and many copies of the DNA have been made, they can also be run on an agarose gel with a size marker, and their size can be determined. So this is another way of actually trying to see how many copies of the CAG repeat an individual has, and there are 2 copies of the Huntington's gene. So we're going to look at another animation here, which also I hope will clarify your understanding and goes into quite a bit of detail about the PCR process.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called TAC polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the TAC polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. TAC polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. TAC polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
There really isn't any way to overestimate the impact that PCR has had on molecular biology, forensics, and several other fields as well. The ability to take a little tiny piece of DNA, one strand, and get to 1 million copies in a 3-hour cycle removed the need to generate large amounts of samples to work with. Previously, people had to really be able to collect blood samples or a tissue sample that was rich in nuclei. And if you wanted to work on something like a flea or lice or anything small like that, you were basically out of luck if you wanted to look at DNA in a single organism, because DNA had to be pulled together in order to get enough to work with. So Carey Mullis really deserved that Nobel Prize, even if he was an avid user of LSD, and really made a wonderful contribution to science. This slide is showing you a picture of bacterial TAC DNA polymerase. It's a beautiful enzyme. I use it every week in my lab, so thank you, Dr. Mullis. And this actually concludes part 2 of online lecture 8. and all of online Lecture 8. And please make sure to review this, and also if you have a laptop and are able to download the KCIT software onto your laptop and bring it with you to class on Wednesday, November 6th.